we're talking about when you really don't have any slides. Okay, so I don't know why I'm the only one who put a picture on there, but they told me to, so I did. But you, I mean, obviously I'm right here. It's really me. It's me giving it. So, and I'm going to talk today about novel approaches for displaying functional classes in RNA-seq. And it's going to be using a type of statistic called compositional data. So, as everybody knows, RNA-seq is RNA sequencing. There's all kinds of different types of RNA sequencing, but at the end of the day, what you're looking for are patterns in the samples that match probes. And there's a lot of complicated bioinformatics that are associated with deep sequencing, where you really don't know where the sequences line up, and so you have to use algorithms and kind of make a best guess. That isn't what I'm going to talk about. I'm going to talk about post all that stuff. What do we do to visualize the data after you've done the bioinformatics for the sequencing? All right. Now, one of the things that I'm going to assume in my talk, because I'll prove to you all in a minute, is that RNA-seq is compositional in nature. And this is an important construct because there are really good mathematical features of compositional data that make different types of analyses easier to do. So I'm going to use one of those constructs today in an example. And that example is when you look at data as compositions, you can group data and summarize those compositions and do math on them without losing any of the original mathematical structure. So it's really a very cool and not necessarily a correct assumption if you're not looking at data in this compositional framework. I'm not going to go into the math of all this, but Aitchison is the kind of the king of all of this, and he has a book and several papers, and he's actually proved this fundamentally, and I just use it. So what do I mean by compositions, right? What I mean by compositions are that we can view, say, those counts of, for each individual, I can view the number of calls for a particular sequence. Within a subject, it has a constraint that all the components are positive, because you can't have negative numbers of cells. That you can think of it within a subject, so you're looking at a vector, and in a way, they all have to sum to a constraint. In RNA-seq, the constraint is the number of calls you have within a subject. So, you know, it's always going to be how deep your sequencing is on an individual. So you can also think about it over a plate, because every sequencer that you use has a finite number of calls based on the technology itself. And so depending on what sequencer you use, you actually have to worry about what we call read depth, right? If you have some sort of a sequencer that has like the highest Illumina sequencer, I think now is a HiSeq, that one has billions of calls available, so you could use a full 96-well plate. But a lot of the earlier MiSeq experiments, you could not. And even if you had a targeted sequencing array, if you will, or a panel, you had to be very careful about the number of wells that you fit up into sequence, because you needed to ensure that every single subject had enough reads to be able to go down to some of those sequences where you might only have 4 or 5 reads, right? So you don't want to run out of reads. It's the basic premise. So that assumption is actually held that within a given subject, you have to have, or you always have, a finite number of reads that are available. So when you have these kind of constraints, I was telling you this is great because if there's some mathematical properties that I can use, but it actually complicates the analyses in some way, because if you ignore it, you could also get spurious results, which Pearson in 1897 said that spurious correlation is fraught with difficulty and danger because, you know, I'm using this constraint or this type of data structure in a way that really makes it useful, but you could get the the converse, where if you ignore it, you could get incorrect results. All right. So, as I've already said, our basis here is going to be the number of calls within a subject. Okay, and that's total over all the different sequences that we could possibly have, whether it's targeted or deep. We also have the total number. Well, each of these is going to be the sequences themselves, and then the summation of each of those are the total number of reads for each case. Does that make sense? Anybody who's been working with RNA-seq data knows this. So the composition here is for every single sequence or every single target or a probe, you actually have how many calls you've had for that probe out of the total number of calls available. And this is all within a subject. So we can think about this as a proportion of reads that are allocated to each of my known probes or sequences. And I can get a geometric mean of all of those, right? Because we know that that kind of works a little better than just simply getting a mean. And I can actually think about this geometric mean. I can think about this as a normalized. I can normalize it, right? So I can normalize this by this geometric mean. So that makes a lot of sense, right? Now, I've done a lot of sort of standardization because I've gotten a proportion. I've made the assumption that each individual sequence can be summarized within a person, sort of as a composition. And then I can standardize within a person, or I can standardize across people, because I can use either of those, and I'm still kind of working in this compositional space, right? So you could always also think of these counts per million or these kilobyte bases, right? That's actually also a composition. That's within a person. That's essentially this thing. Right? So this is a counts per million. This is what we call a centered log ratio. So they both have different properties. They're both compositions. So the mathematics of them, it's pretty interesting that something that people always use, which is this counts per million or this reads per kilobyte, are actually compositions. So you're working in the compositional framework, whether you realize it or not. So now let's talk about one of the awesome properties about working in this space, is that I can actually think of all of my data as in hierarchies, right? And what I can do is I can sum over all of those hierarchies while still maintaining this nice mathematical property. So I've got total aligned reads as my first kind of hierarchy. I've got a bunch of samples within that, right? I've got different groups within those. And then I've got all of my sequences, okay? So you can see the hierarchy in the data, you can create the hierarchy, and you can normalize by all of those hierarchies. And all of this is still within this mathematical framework, having all of these nice mathematical properties of compositional data. So I'm going to show you an example and then show you how you can use these properties, okay? So I had extracted RNA from 32 pancreatitis samples, okay? These are kind of, these are not cancer, but they're not normal, right? And then I had 32 normal pancreatic samples. And we used Illumina, MySeq, and I want to use as my kind of grouping level, I want to use Go categories. I want to use a functional category because I know that there are different probes that actually have functional characteristics, and I really want to see if there's differences in those functional characteristics between these two. So I'm not looking for individual probe differences between these two groups. I'm looking for functional differences. I'm using fancy terms because if you're in compositional space, you're actually working on the simplex. So all of the beautiful things and beautiful geometry that happen on the simplex. So you'll have to read Aitchison paper. Believe me, it's gorgeous. I mean, it's really pretty. It's great math. But I'm using this simplex kind of, I have all of these, I have, if I needed to, I could prove that all of this is correct. But here I'm just going to show you in 10 minutes how this works, or how it can help our lives, and more importantly, help us interpret data. So the first thing I do is I just do a biplot of my two groups. So my blue dots here are my normal pancreas group. My red dots are my diseased pancreas group, okay? And each of these stars off of my biplot are actually functional categories. So you can see this one is a cell cycle functional category. This is an NK cell function. This is a type of tumor microenvironment kind of functional category. I have adhesions. You could see that there's quite a few that are kind of clustering up here in the adhesion group of the diseased pancreas, right? So there's something going on with that functional category in this group. And then here we've got a lot of immune function, right? So that maybe makes sense if you have a well-functioning pancreas that it's doing what it's supposed to in terms of immunity. And then when you're not, you've got all this other stuff happening. So we're maintaining all of this. Now what I did was, first I want to look to see the differences of how these things are actually, where these two different sample types are kind of clustering by go category. But then if I really want to know what's going on, I'm going to use my geometry to actually look at genes within that category. So now I've highlighted 2 or 3 different categories, or go categories that I want to look at. This one's the cell adhesion, because it was obvious that we had something going on there. So let's look and see which of the genes are associated with that go category that are explaining this clustering kind of mechanism that I'm seeing. So all of these now, same kind of idea, same concept, but now I'm grouping instead of by functional category, I'm now grouping by gene within a functional category. I'm still maintaining my compositional structure, and I can actually look to see which genes are kind of driving these differences. Okay, and I can show you on a star plot, or I can actually look at the average of these reads, right? And I can do this on a transformed scale that I talked about before, this centered log ratio scale. Part of the reason historically, just so you know, in the compositional literature, people like to use the centered log ratio scale because you can use centered log ratio you can use a logistic regression on that. It's just a transformation, but it makes it very easy to use sort of typical modeling strategies, if you will, if you're working in this particular transformation space. So, but for us, it's just we can actually look at each one of these genes and look at the proportion of reads that's associated on this transformed scale. And we can see this is one of the big ones that's driving this, is this NF1 gene. And it's a very highly, proportionally, it's very high in the diseased pancreas and low in the non-diseased pancreas. And you can also see the patterns of highly expressed and relatively lower expressed compared to average. Okay? So I also did the same thing in this immune function probes, right? Because that was the other one that we seemed to, in terms of our biplot, we saw that there was a lot of the normal pancreas that seemed to be clustered out at that particular category. And here we can identify which genes are likely to drive that. So basically, first thing is that we actually can look at RNA-seq as compositional data structure. And the reason that we like to do that is because there's actually beautiful very elegant mathematical properties. They're also pretty well established. we can actually think of a lot of the transformations that we already use as compositional in nature. Anybody familiar with quantitative RNA-seq? That's also compositional because you basically are normalizing to a housekeeper. That's the same kind of a transformation. And then once we are in this space, we can actually use this compositional geometry to actually as a multivariate feature. So I didn't show any of that. Right now I was mainly in the univariate space, but the one positive thing about being in this compositional geometry is that the multivariate works a lot better. So I'll tell you, this compositional structure doesn't help you in these kind of like gene expression profiles where you're looking at between two groups. It really doesn't matter. Where it matters is when we're looking at this multivariate framework or when we're trying to interpret things. And in those two cases, like when interpret kind of a larger scale interpretation of the data, in those two cases, if you don't look at your data in this compositional framework, you could lose, not only could you lose information, but you might actually get spurious results. But not probably in that univariate, so you don't have to worry about that if you're just doing the kind of pairwise two group or four group, whatever comparison. Okay? All right. Thank you. Whew. Sorry, you guys. My slides weren't loaded. It was not fun. Wasn't fun. But we worked it out.
Someone mind timing for me? Letting me know when I've been talking for 9 minutes.
So what was your name? Are you giving the next
Yes,
talk?
I am.
So what was your name?
Christina Riemer.
So I don't know if your slides are on here.
My slides are on the computer. I put them on there.
Oh, okay.
Yeah, sure. I just want to go.
2:30.
Well, I'm supposed to be in the panel at 2:30, so
2:28.
yeah.
That'd be awesome.
So it should be. Hi everyone, my name is Christina Riemer and I'm currently working as a scientific programmer in the Data Science Institute here. And today I want to talk a little bit about the process of producing scientific software for scientific research. And so I'm just going to start off with this analogy, basically using containers that are used to hold food, and have sort of this spectrum here where on the left end we have your typical like styrofoam to-go containers. They're something that you use one time and you discard when you're done with them. You can't use them for many other things besides the one purpose. On the other end of the spectrum, we have glass Tupperware, which different than the to-go box is that you can use it many times for many different purposes, and it has way fewer limitations to it. So to relate that to scientific code, most of the code that is used for scientific research is this styrofoam to-go container. You can really only use it one time for a single project. And that's fine. It's better than nothing because you can use this code to get whatever results you need for a manuscript or a report or anything like that. And also, it's better than all the other alternatives because you have at least some of your tuning and analysis steps recorded somewhere in your code. But this is also not a very good thing for science because then it means that you can't later on prove that you did what you think you did. You can't repeat the work that you've done in the past. And so how can you really believe that whatever you did is what you think you did? And so in order to make your code more believable, you have to try to make it more reproducible. And so this right here is just a screenshot from a project from my PhD that I worked on, and I tried really hard to make that project more reproducible. And so what I mean by reproducibility here is that you can take your raw data and your code, give it to someone else, and they should be able to run all of that and create exact copies of your analyses and all your visualizations for the work that you did. So I tried pretty hard to do that here. But one of the difficulties with reproducibility that can discourage people is there are so many things you can think about in order to make your work more reproducible. You can think about things like file naming and file structures. thinking about commenting your code well enough to actually understand what each part of it is doing, really tracking the inputs and outputs to a project, and providing documentation for that, and also creating your code in a way that it can be run across different operating systems, and takes into account all of the packages and languages that someone has used for a project. And so this is really difficult to do. And I tried to do it on this project here. I'm not entirely certain I could just pick this back up and run it and get all the same results again. But because I put in all that work to try to make it more reproducible, it would be much easier to get to the point where I could redo everything that I'd done before. Okay, and so the next step after making your scientific research more your scientific code more reproducible is then actually producing software tools for scientists to use. And so this, I'm now learning, is a pretty big step forward. It takes a lot more work because not only do you need to think about all these reproducible principles and approaches, but you have to do some additional things. So you really have to provide super good, clear, easy to understand documentation for a tool in order for people to be able to use it. You have to have a nice user interface that is intuitive, hopefully. And one of the important things that differentiates normal scientific code from actual software tools is that you need to make it make a tool that's flexible enough that it takes different inputs so that you can attract users to your tool. So a lot of scientific code, it just has one purpose. You give it this set of data, you get these results. For scientific software, you actually want users to be able to specify whatever they're interested in as input and get their desired output. And so that requires a lot more work to make your tool flexible. Produce your last Tupperware. Okay. And so right now for my current job, I'm working with this software tool that is still being developed called Pecan. And that stands for the Predictive Ecosystem Analyzer. And basically what this tool does is it runs ecosystem models in a Bayesian framework. And so the idea behind this tool is that anyone should be able to come along, pick it up, plug in whatever model they want to use, and then what we're doing is providing all of the Bayesian framework. So generating prior distributions and producing uncertainty analyses once the model has been run. And so this is a cool, ambitious, really helpful tool that should really facilitate other scientists to be able to do their work faster and easier and in a more replicable way. And so I just want to talk really briefly about two tools that we're using that help make our software tool more usable. So one of the first issues you have to deal with, especially with software tools, is that they require lots of dependencies. So any software tool is going to require a lot of different packages, maybe different languages, certain platforms. And so this is referred to as dependency hell. And so we are using, and I didn't make that up. One of the tools that we're using to help facilitate dealing with that problem of dependencies are containers. So Docker containers and Singularity containers are the well-known ones right now. And so within our Pecan, with our tool that we're developing, we're in the process of taking all of the models that we can currently plug into Pecan and putting them into Docker containers. And so what these containers basically are is you take all the dependencies for a project or a tool, you put it in this nice little unit that's called, well, for Docker, it's a Docker image. And you should be able to just hand that to someone else. They can run it on their computer, and then they can use your tool from within that container because you've already set up all the dependencies. And so they don't have to waste the hours and hours you can spend trying to install programs on your machine. So containers are awesome. Another thing that's really important to provide for a good software tool is documentation for the users to use. And so this right here is a screenshot of the documentation for Pecan. And what we are using for our documentation is an R package called Bookdown. And so what this is basically is that each of our pages, is that it? Okay, I'm almost done. Each of our pages is an R Markdown file. And so our Markdown files combine text and code. And then Bookdown is what we use to basically paste all those R Markdowns together into a website, and then we can specify the layout. And so this is a really, really useful tool. It's easy to work with, it's easy to update, and so that's very important as people are changing the code base for a software tool. You want to be able to have them update documentation easily so they're in sync. And so yeah, this is one of the other things that we're using. And just to say, there's lots of things that you can do to make your work more reproducible and usable, but don't feel overwhelmed, and every single thing you learn is helpful. So, thank you. Sorry for a little bit. But if anybody has questions, email me or tweet. tweet at me or whatever.